Hi, this is Anna, and we're going to be talking about the accessory organs of the digestive system. So this is part 3 of Unit 11. All right, so you have your obligatory notes page right here. So make sure you know all of that information. Let's go on and start looking at pictures. All right, so starting with our accessory organs, let's look first at your liver. The liver is this really ugly thing tucked underneath your ribs on your right side. So take your hand and just kind of push right underneath your rib cage, and you're going to be pushing on your liver. And if you go to the doctor and they're doing a clinical exam, they're going to palpate for your liver to see if it's enlarged or not. So that's one of the things that they're doing when they push on you, is checking out the size of that liver. Now, there's a ton of stuff that the liver does. The liver is an incredibly important organ. So I think most of us kind of think of the heart as like the heart and the brain is the thing that's going to kill you if they get damaged really quickly. And then you can kind of follow that up with the lungs, except you've got 2 lungs. So if one of your lungs collapses because you got a bullet hole through it, you're still going to be able to breathe, okay? You're going to be coughing up blood, but you're okay, really. That can be fixed. The liver is one of those other things that if it gets trashed, you're dead, okay? If the liver function shuts down, whether from disease or from a blunt trauma or from knife trauma or a gunshot, you've got about 24 hours to live. Liver is incredibly important. It's just not going to kill you as fast as a bullet through the heart, okay? You will breathe a sigh of relief in that we're not going to be memorizing everything with the liver. I don't even care if you know right lobe, left lobe. I want you to know location. I want you to know that the gallbladder is going to tuck up underneath it. And then that basically right over here, so let's look at this side, you can see the indentation of where your kidney is going to go, okay? So I want you to associate your right hand with the liver and your spleen with your left hand. That will help you keep you from accidentally flipping their locations. Unless, of course, you've got one of those rare people where the organs all flipped from right to left, which does occasionally happen. Okay? So let's just remember that. So you've got the liver. You need to know the gallbladder right here. And I want you to know the duct work that's going to be coming off. Okay? So remember we've already studied the hepatic portal system. So you've got the hepatic portal vein coming in. Okay? So that's going in right there. And you've got your hepatic artery. All right? So that's right there. Those are going to go into the liver. Draining the liver, you are going to have the hepatic veins, and those hepatic veins are going to drain into the inferior vena cava, okay, which isn't really being shown here. What else? You can see the bile duct coming off of the gallbladder as well, okay? Let's move on. All right, so this part you do need to know. You need to know the histology structures of the liver, be able to identify it on a model on a drawing, and to a lesser extent the photomicrograph. I'll talk about what I want you to know for the photomicrograph. All right, so looking at these structures, let's start right here first. Let's switch to yellow with your central vein, okay? And then actually, let's not start with that first. Let's actually start with the hepatic portal vein. So that's going to go in right here. And then you're going to have flowing of blood coming from the digestive system in these directions where they're going to then flow into your central vein. Okay? So that's the direction of blood flow. Now, this blood right here, all right, this is full of nutrients, but it's low oxygen, okay? So you also have to have a hepatic artery bringing in oxygen to those cells. Now the hepatocytes, so those are called, all of the liver cells are called hepatocytes. Those are going to be taking nutrients from the venous blood, but getting oxygen from the arterial blood. Now, let me erase some of this. All right, this is your hepatic triad. which is going to consist of the hepatic portal vein, the hepatic artery, and the bile duct. Okay, so this is going in, this is going in, this is going out, okay, in terms of the flow of stuff going on. Now let's look over here at these little hepatocytes. Okay, so these are all hepatocytes. All right, in between them you're going to have little bile canaliculi. So where do they have that labeled? Right there. So you can see them cut transversely here and then going longitudinally there. In the hepatocytes, you're going to be synthesizing the bile salts, which are going to drain into the bile canaliculi. So here's some more here, here, here's some right here, right there. Here's some coming out. And you can see that those are going to go and drain into the bile duct, which is going, the bile ductule, meaning it's smaller than a bile duct. All right. And those are going to drain out of the liver. Now, the entire structure, you can see you've got this basically kind of lobe-like structure going on. Oop, where did it go? There it goes, right there. And you can see you've got the hepatic triads in the middle, in between all of these lobes of the liver. All right, let's look at the next slide. All right, so right here we're blowing up the hepatic portal triad, okay? And you can see now they've got these nice arrows showing the directionality, all right? You can see the hepatocytes a little bit clearer, and then you can see the little canaliculi here and there. They're showing you kind of draining and going out into the bile ductule, okay? Now, these sinusoids are going to be collecting this venous blood going this way, all right? So those are called sinusoids. Those sinusoids all empty into that central vein, which is going to go to the hepatic veins, which is then going to go to the inferior vena cava. Okay, one more thing I want to point out, let's pick a different color, are these cute little cells right there. Okay, so those are going to be your Kupffer cells, which is basically a type of dendritic cell, and they're going to be monitoring the area for pathogens. What else can I say? Oh, sometimes people call, so dendritic cell is also, not always, but often a type of macrophage. In this case, it is definitely a macrophage. There are other dendritic cells that arise from a different line than the macrophages, and you will study that in pathophysiology. All right, so here we are looking at a low magnification photomicrograph. And what you're going to notice is the lobe structure right here and here. And you can see the stroma tissue, fibrous connective tissue separating. It's a very thin fiber. Don't worry about it. Just know that you can see the variation. You can, of course, see your central vein right here. Okay. Then what you know is you've got a lot of hepatocytes in here. And that's kind of what you can see, and you can't really see anything else. Okay. Now, in between all of these lobes, you have a really nice view of the portal triads, and you can see the vein and the artery. So notice the thin walls of the vein. All right, that's telling you it's a vein. But right here, if you remember back from when we studied this, the tunica media is much larger on the arteries, so the hepatic artery is very distinguishable from the hepatic portal vein. Okay. And then the thing that doesn't look like an artery or a vein, that's your ductule. All right, next slide. All right, so that was it for the liver. So now we're going to look at the pancreas, and now we're going to also look a little bit more closely at the biliary network. So that's the network that goes with the bile. So right here, you've got your gallbladder. All right, continuing on, the gallbladder. So what I need you to memorize is the duct work. All right, so you've got the cystic duct, right? Ooh, let me change color, right there. All right, that is coming out of the gallbladder. All right, so this is going to be the direction. Actually, let's ignore direction for now because it changes. All right, we've got coming out of the liver the hepatic ducts. Now, this one you got to be careful with. Look at the word common. Okay, you've got lots of hepatic ducts. You've got one common hepatic duct. You've got to put the word common in there, or it's not the right name. Okay, now the common hepatic duct and the cystic duct will drain into the bile duct, okay? The bile duct comes up here, and it's going to merge right here, coming down the center of the pancreas is the pancreatic duct. The pancreatic duct is collecting the digestive enzymes, okay? Also bicarbonate ions. So that's going to be your lipase, your protease, protease, your amylases, all of those things. And they're going to come into this common area called the hepatopancreatic ampulla. So hepato, liver, meaning it's kind of bile, and pancreatic, it's picking up those digestive enzymes from the pancreas, all right? Now, when there is a reflex that relaxes this sphincter made out of smooth muscle, this is called the hepatopancreatic sphincter, then this stuff can flow into the duodenum. Now I want you to notice we have one more duct coming out of the pancreas, the accessory pancreatic duct. There is no true sphincter there. It just drains passively as things move along. This one drains a lot if you have eaten something and there is a spinal reflex causing, or a locally acting reflex that causes this structure to relax. Okay? Now, the way this works, and you'll study this more in lecture, is the liver makes bile. Gallbladder stores and concentrates bile. You've got to memorize that. You've got to know that. Okay, that will be a question that you'll see on the quiz or midterm or final. Not midterm, final. All right? So if you are eating something with lots of fat in, so you're having a milkshake, okay? The bile is gonna come down these, and it's gonna go into here, and it's gonna drain directly in here. Now you're drinking 2 milkshakes. You don't have enough coming out of the liver fast enough. The gallbladder will contract and send stuff down as well, okay? So that you're getting even extra bile put into there. Now let's switch it up. You're eating a salad, okay? You're eating a salad. There's no fat in it. This doesn't open. Stays closed, or it only opens a little bit to get some digestive enzymes in there. Or let's say you're fasting. You're going to be doing one of those intermittent fasts where you don't eat from 8 p.m. to 8 a.m. So let's go with that. We're not eating salad. We're fasting. Your liver is still making bile because that's what it does. It's going to come down here, and it's going to hit a wall. And it's basically going to start backing up, and as it backs up, it's going to go into the gallbladder where it'll be stored and concentrated, and then when you come off of your fast, then you can use it. All right, I think that's everything I want to say about this slide. Actually, one more thing. What I want you to do is I want you to visualize the stomach, which is going to be basically kind of right here, hiding all of this structure. And then remember the stomach will have, actually I kind of drew this, it's more like that. And then right here is where you're going to have that pyloric sphincter. Okay, next slide. All right, this is a schematic drawing or a flowchart drawing or a concept map drawing, whatever you want to do, that shows you the flow from the liver and gallbladder down to the duodenum, okay? And this is demonstrating if you're actually using the bile, okay? Remember that if you don't use the bile, it basically will back up and go into that direction, okay? And then if you're using it, it goes into this direction. Okay? All right, next slide. All right, so let's start looking at the photomicrographs. So right now we're looking at the pancreas low magnification. This is coming from A&PII. And you should be able to see this, and the first thing that's really going to jump out is going to be these things, which are hard to tell that they're there right now because of the way it's stained. But I've got all of those things, and those are your pancreatic islets. So here, they're staining islets. Here, they're staining the acinar cells, also called the acini. Okay, so it's showing you the contrast. So the bulk of the pancreas is making digestive enzymes. Okay, whereas the islets are making glucagon and insulin. Okay, next slide. All right, so here we're kind of blowing these things up so that you can see the structure of the acinar cells. So they're taking on this nice bulbous shape. They're making your enzymes, which go into a duct, which is then going to drain into the pancreatic duct. Okay. Here you can see, so let's move over to this. You can see the simple cuboidal epithelium of those acinar cells, so you should be able to identify that these are simple cuboidal. Okay? All right, let's do the next slide. All right, this slide is really highlighting going in at higher magnification. So this is medium mag, and you can see the acinar cells there, but you can also see really nicely the staining difference between the islets of Langerhans. You can see that there's a little moat right here. You can see the moat right there, okay? And so they pick up the stain differently, okay? So you should be able to recognize that. Do not confuse with kidney glomerulus. That will be one of these weeks' histology exercise where you have to do a compare and contrast of these two structures. You don't want to accidentally confuse them. All right, let's go on to the last slide. And here's your last slide. It's very easy and quick. Spleen, largest lymphoid organ in the body, located posterolateral to the stomach under the left side, okay? It is not part of your digestive system, but we often talk about it because it's kind of tucked up there with those digestive organs, and it's easy to see now, and you want to know its orientation, okay? So that was the end of this particular video lecture, so we'll stop there.